Okay, we'll get started. We're going to try to finish up the chapter on photosynthesis. Last time we had talked about how, actually we'll just call this the electrochemical gradient continued. Last time I kind of left you hanging, but I told you that we were able to show that 12 moles of protons across the thylakoid membrane created about 200 kilojoules of energy. So remember that calculation where I had delta G, and then I had some term here, RT ln, and I had a concentration. I think this was, let me see, if this goes in to out, then this is going to be in to out, and then plus ZF membrane potential going from in to out. Okay, and we had said last time that experimentally there is almost negligible amount of electrical component to this equation, and almost all of the energy is derived from this concentration gradient. And I explained how there appears to be not a great net difference in negative ions versus positive ions across the membrane, but there is a great difference in the concentration of the protons, a differential concentration of the protons. Okay. It turns out that we get about 3 moles of ATP from these 12 moles of protons that get pumped across the thylakoid membrane. And so you could see you could actually calculate the efficiency, so the percent efficiency of photosynthesis in terms of converting proton motive force into chemical energy is going to be equal to 3 times 30.5 kilojoules per mole. Actually, the moles don't even count here anymore because we've taken out 3. Okay, it's 3 divided by 200 multiplied by 100. Take the absolute value just to make sure that all turns out to be positive, and that's going to come out to be approximately, let's see here, 91.5 divided by 200 times 100. What is that? About 48%. About 48%, I guess. Let me get my calculator. I'm just guesstimating there. 91.5 divided by 200, 46%. So you get about 46% efficiency in this system. Okay. In terms of converting the proton gradient into actual chemical energy. Okay. I want to talk about these experiments done by Jagendorf and Uribe back in 1966. And what they did is that, similar to what I had talked about before, they created they actually created a gradient across the thylakoid membrane by taking chloroplasts. So here's a leaf. Okay. Then they isolated the chloroplasts. And they would take those chloroplasts and they would put them in a buffer. So we'll just take a little beaker here, and here's a buffer. And they would have, the pH of that buffer would be pH 4. So here's our buffer, here's our chloroplasts. Okay, so they would put that into a buffer of pH 4. And they would wait a little bit of time, and after a while, the pH inside the lumen I'll just sort of blow up a thylakoid membrane. So here's a thylakoid. Okay. And so what's happening is that as they soak in this pH 4 buffer, some protons seep into the inside, and so the pH becomes 4.0, okay, as well as on the outside too. So the pH will be 4.0. And then what they do is that they just centrifuge the mixture of chloroplasts with the buffer. So they centrifuge. Okay. So you have all your chloroplasts down here as a pellet. So we're going to pellet the chloroplasts. And then remove the supernatant. And then what they did is that they put the chloroplasts into a new buffer. And this buffer is pH 7.0. And they also put in ADP plus inorganic phosphate, which they labeled with P32, so it's radioactive. Okay, and then they would wait about 15 seconds. And they would measure the ATP with P32 incorporated in it. They would measure that level. And sure enough, what they would find is that they were able to produce the ATP. Okay, so they're able to produce that in the system. So let's sort of analyze this at the level of what's happening at the thylakoid. Okay, so here's a thylakoid. We have a lot of protons on the inside in the beginning, and then when we shift to pH 7, we have pH 4 momentarily on the inside. We've got pH 7 on the outside. So where are the, relative to, how can I say this? Basically, there's more protons on the inside than there is on the outside, so I'll just write a little tiny h out there. Okay, so there's a small amount of protons on the outside, large amount of protons on the inside. And of course, you've got your ATP synthase here, ATP synthase. Okay. And the question is, can you produce ATP in this system? Okay, and we are producing the ATP because the protons are going through that ATP synthase, and they're actually able to create ATP. So it's a beautiful system, very simple idea, using very rudimentary tools to demonstrate that you do this pH gradient thing. I'm sure they did some controls. I'm sure they switched it so that they would do the initial experiment in pH 7 buffer, and then they would resuspend in pH 4 buffer, they probably don't get anything. They shouldn't get anything in that case because you're not running the pH gradient in the correct way. Another control that they did, which is important, is you could put in an uncoupler. And we've talked about uncouplers before. Does anyone remember the name of one uncoupler? 2,4-dinitrophenol. Very good. So I'll just call it DNP. That's an uncoupler. Okay. And you can put that into the system. And of course, that will dissipate the pH gradient. And so now you are unable to drive this reaction. And then they wanted to figure out whether or not they could distinguish between an electrical gradient or a chemical gradient. And the way they did this is they used something called an ionophore. Ionophore. What are these ionophores? These ionophores are organic molecules. They have kind of a cage-like existence. And what happens is that they can trap ions of a certain size inside them, and they can transport these ions into or out of or across membranes. And they use an ionophore that was very specific for potassium. So the potassium would get caught into this ionophore. This is an organic molecule. And so they found out that they could shut down any type of membrane potential by putting in this ionophore, and they would put a little potassium in it. And what would happen is that the potassium would end up being on the outside here, okay? So you get a lot of potassium on the outside, very little potassium on the inside, okay? And so this would basically negate the membrane potential, okay? So there's no, so basically this is going to be, this delta membrane potential is going to be 0 volts, okay? Let's be very clear about this. Without this ionophore, you're going to have a buildup of positive charges on the inside and negative charges on the outside, okay? Because protons are positively charged. But now, if you put in potassium and you create a large amount of potassium on the outside and a small amount of potassium on the inside, that's going to negate that charge difference. So the membrane potential now will be 0 volts. Okay. So what do you guys think? Do you guys think that you're still going to get ATP synthesis here? Someone's shaking their head yes, but barely. I can barely perceive it. Why? Why do you think you're still going to get ATP synthesis? ATP synthesis. It's true. You will get ATP synthesis. But why?
Because it wouldn't come clear?
Excuse me?
Because of the DNP?
We're not putting any DNP in there. So there's no DNP. That's a separate issue. Yeah. If you put in DNP, for sure, you do not get ATP synthesis, right? Because now you basically equalize the proton concentration across the membrane. I'm talking now about the ionophore. Anybody want to guess? Why do you still get ATP? The answer is you still do get ATP synthesis, but why? Anybody? Well, we could wait a little bit. That's okay. Just think about it. It's not an easy question, actually.
So because there's still some charge difference?
No, there's no charge difference. And remember, experimentally, I told you from the very beginning that when you look at that equation, the membrane potential that was measured to be 0 when you use plant chloroplasts. Yeah?
Is it because there's still a high concentration of protons inside and a low concentration?
That is correct. You still have the chemical gradient. So I'm trying to attach meanings to these words. You have a chemical gradient, but you don't have an electrical gradient. Okay? So because you still have the chemical gradient, you're still these protons still want to go to the opposite, want to travel through the ATP synthase in order to equalize that concentration difference. Okay. In mitochondria, you actually have more of a charge difference which runs this reaction, whereas in chloroplasts you tend to have more of a concentration difference. Okay. So that actually, I think that's a good lead-in to this one sapling problem, which I don't think is very clear, so I'll go over it with you. And it's one of these where you're supposed to put these different terms in these little bins here. So one of them they call photophosphorylation. The other category is oxidative phosphorylation. Oxidative phosphorylation. And the third category is both. Okay. And you're supposed to put these terms into the different bins here. And what are these different terms? One of them is quite easy. Occurs in chloroplasts. Okay. Occurs in plants. Another one is occurs in mitochondria. Another one says involves a smaller electrical component. Involves a smaller electrical component. Another one says involves a larger electrical component. Produces ATP. And then this one is involves a proton gradient. Okay, we'll just go ahead and number these. 1, 2, 3, 4, 5, 6, 7. Okay. So let's think about this. 1 occurs in chloroplasts. What do you guys think? Photophosphorylation, oxidative phosphorylation, or both photophosphorylation and oxidative phosphorylation? You guys did this, right? This is sapling learning? Okay, all right. What do you guys think? Someone said both. Okay. Photophosphorylation occurs in chloroplasts. Yes, so that would be one. Okay. Okay. For sure, it would be photophosphorylation. The question is, do you also do oxidative phosphorylation in the chloroplast? And we talked about how the phosphorylation occurs in the chloroplast, right? Phosphorylation occurs due to the proton gradient, but how is that proton gradient actually created? Yeah, it's caused basically by photosystem 1, it's by chlorophylls, right? And so we don't, that's actually a good point. We don't classically call that oxidative phosphorylation, okay? We don't call the production of O2 from water oxidative phosphorylation. So, although to be honest, we are getting, well, we're not using oxygen as the terminal electron acceptor. So, anyway, the answer is one, that occurs in chloroplast, photophosphorylation. Okay. We can cross that guy out now. Okay. Occurs in plants. What do we got? Photophosphorylation, oxidative phosphorylation, or both? So the question is, does photophosphorylation, oxidative phosphorylation, or is it just photophosphorylation that occurs in plants? What do you guys think? It's going to be both. Very good. Why do you think it's both? Why do you guys think it's both? Why do you guys think that photophosphorylation and oxidative phosphorylation both occur in plants? What gives you that idea? Okay, plants have what organelles? Okay, they have chloroplasts for sure. What other organelles do they have? Mitochondria. They also have mitochondria, right? So that's the reason why it's going to be both here. Okay. Okay, this is an obvious one. 3 occurs in mitochondria. What do you guys think? Oxidative phosphorylation. Okay. I guess I should try to make these look the same. Okay. Okay, involves a smaller electrical component. Okay, photophosphorylation, right? It involves a smaller electrical component. So that's going to be right here. Okay. That's driven more by a chemical gradient. I just talked about the ionophore experiment. Okay. Involves a larger electrical component. It's going to be occurring in the mitochondria, and that's going to be oxidative phosphorylation, right? And if you look back at the calculations we did when we calculated the electrical component and the chemical gradient component, it was the electrical component that was larger in the mitochondria when we figured out the amount of energy from the protons across the membrane. Okay, so we did those two. Okay, produces ATP. Both, right? Photophosphorylation as well as oxidative phosphorylation will produce both. Very good. Okay. 7 involves a proton gradient. Both. Very good. Very good. All right. Okay. We've talked about the reactions in photosynthesis that require photons. We talked about it in the Z scheme, and we also talked about it in the cyclic photophosphorylation scheme, or cyclic cyclic photophosphorylation scheme. So I've given you examples of that. And remember, the cyclic one, that occurs when you have sufficient amounts of ATP, sufficient amounts of NADH, but now you want to make ATP. We didn't talk about the dark reactions. And the dark reactions are those reactions that lead to the incorporation of CO2 into sugar. And basically, you're going to reduce the CO2, and you're going to actually create glucose from this. Okay? So we'll talk about the dark reactions now. So for the dark reactions, CO2 is eventually going to be made into glucose. Okay, your book actually, this is a pretty complicated process. This is called the Calvin cycle, by the way. After Calvin, who figured this stuff out in the 1940s, using radio-labeled CO2. So he used radio-labeled CO2, and he kind of followed where the CO2 went to when he incubated them with some organisms that had chloroplasts in them. I think some algae. Okay. And then what he would do is that he would stop the reaction after a certain amount of time, and then he would separate all the components on paper. And then he would expose the X-ray film, and he would see where the little spots were, and he knew what those spots related to, what chemicals those spots related to. Oh, you know what? Before I go into this, no, I'll do this first, then I'll go through some problems. Okay, I'll do this first. So, let's see. Your book does a pretty good job of describing this. I'm going to just go through this pretty much on a basic level. There's some good pictures here that I want to show you. So this is on page 695 of your book. Okay. And this is figure 1623. And we have something called two stages. One's called acceptor regeneration, and the other one is called CO2 fixation and sugar production. Let's just talk about this one. This is easy to understand. We're getting the CO2 into some carbon molecules, and we're going to create sugar from this. Okay? And so what happens is that you have this ribulose 1,5-bisphosphate. And I think the easiest way to understand this is to write down the number of carbons we're talking about. So this is this is a 5-carbon molecule, this ribulose. Okay? That's 5 carbons. Obviously CO2 is 1 carbon. Okay? And then, does anyone remember 3-phosphoglycerate? How many carbons does 3-phosphoglycerate have? Think about it. Sounds like glyceraldehyde. How many carbons are in there? Someone said it. Don't be shy. It is 3. Very good, 3. So we're actually going to get 2 times 3 of these 3-phosphoglycerates. Okay, 3-phosphoglycerates. This is actually a 3-carbon sugar. So 2 times 3 plus equals, that's going to be 6. That's going to be equal to 5 carbons here and the 1 carbon from the CO2. Okay? And then here we've got 1,3-bisphosphoglycerate. That's also going to be 2 times 3, because you're going to get 2 of these molecules. Okay? And then we come to one of our old friends, one of our familiar friends that we had earlier in the quarter, glyceraldehyde 3-phosphate. Where have we heard about glyceraldehyde 3-phosphate before? Glycolysis, yes, glycolysis, as well as gluconeogenesis, right? So it's going to be also part of gluconeogenesis. So glyceraldehyde 3-phosphate. And we're going to get 2 of these 3 glyceraldehyde 3-phosphates. And you can imagine how these glyceraldehyde 3-phosphates can come together and eventually form hexose. That's going to be a 6-carbon molecule, okay? And you're going to make these hexophosphates, and eventually you're going to make these polysaccharides. And the hexose phosphates you're going to make is actually glucose. I think it's 1-phosphate or something like that. Glucose 1-phosphate. Okay? So basically, from here on out, you're familiar with this because this is gluconeogenesis, started from glyceraldehyde 3-phosphate, which you remember. Okay. However, some of this glyceraldehyde 3-phosphate comes back and creates this acceptor molecule called ribulose 5-phosphate. That's 5 carbons again. And then these 5 carbons, this ribulose 5-phosphate gets another phosphate from ATP, and you make this ribulose 1,5-bisphosphate. Okay, so I think it's fairly clear that this carbon is getting fixed into some other carbon molecules, and eventually you're going to go through gluconeogenesis, and you're going to create this hexose, and you're going to go even further. Remember, when you have a large amounts of glucose around in the body, you make, what did I say it was? Glycogen, right? But plants, they make something a little bit different. It's called amylose, okay? But it's just a polymerized form of glucose. What else can we take from this in terms of big picture items? Well, you could see that we're consuming ATP, and we're also consuming NADPH. The consumption of ATP tells you that this is what, a catabolic pathway or an anabolic pathway? It's consuming ATP. Anabolic. This is an anabolic pathway. And also, is this going to be a reductive pathway, or is it going to be an oxidative pathway? In other words, are your final products going to be more reduced compared to your substrates, or are they going to be more oxidized? Are they going to be more reduced or more oxidized? It's going to be reduced because this NADPH is going to donate what? Okay, let's get very specific. I know it's going to be electrons. Hydride. Very good. We've talked about hydrides before, right? So imagine now we're using NADPH. And of course, the cell keeps the ratio of NADPH to NADP plus high. Okay, so that's sort of the big picture item here that we should consider. Okay. This enzyme, by the way, that incorporates the CO2 into ribulose 1,5-bisphosphate, we have an abbreviation for that, and the abbreviation is called Rubisco. Rubisco. Let's see if they even tell you what that is here. Oh yeah, here we go, Rubisco. So on the next page of your book, here is this word Rubisco. And you should just remember this word, Rubisco. That is a synthase. It is a synthase, and it is the most abundant protein in the world. The most abundant protein in the world. As a matter of fact, I think for every human being, there's like 20 pounds of this protein on Earth. And it's not in humans, obviously, it's in plants, but there's a lot of this protein hanging around. Okay? I think it makes up like 15% of all the protein out there. Just this one protein, Rubisco. Okay, so since it's such an abundant protein, it's an enzyme, I think we need to cover a little bit of its mechanism, although it's one of these really complicated ones. Almost like the water splitting complex, it's one of these really complicated enzymes that scientists have been trying to work on for a long time. Imagine if you could just like synthesize a lot of this stuff and have it work. You could basically start to lower global warming, right? By like sucking out all the CO2 that's being produced in these factories. So scientists are like dying to figure out how this thing works so well. Okay, and we don't know. There's a lot of sketchiness to it. Okay, so we'll just talk a little bit about the the mechanism here. Okay, so this is Rubisco. So here's the substrate. You got this base. It's going to take off this proton from this number 3 carbon. And then you have this other base that's going to take off this other proton here. I'm not sure why they didn't put that in here, but you can take off this other proton here. Okay? And eventually, you're going to attack the CO2. And that CO2 is eventually going to end up in glucose. Okay? So that's the CO2 that's going to end up in glucose way further down the line. Okay? And then finally, you're going to have water. Water is going to come along and cause a nucleophilic attack on this 3rd carbon. So a lot of the action's on this 3rd carbon. And then you're going to get cleavage right here when you have this negative charge is going to make a double bond carbon. So you're going to make a carboxylic acid out of this. Because this reaction, you're cleaving a carbon-carbon bond, we actually call this a lyase. Okay? Like a lyase right here. Okay? This part right here is a lyase. Okay? And so finally, we're going to get 3 carbons up here, 3 carbons down here, and both of these will make 3 phosphoglycerates. And as I told you before, we're going to have 2 of these. 2 of these molecules of 3 phosphoglycerate. Sorry, that's not coming in here because of the light. Okay. This will eventually be reduced to 3-phosphoglyceraldehyde. Okay. All you have to do is create an aldehyde out of this. An aldehyde is more reduced than a carboxylic acid. Actually, those reactions are shown very simply down here. So here we go. Here's 3-phosphoglycerate. In order to make this oxygen a good leaving group, we phosphorylate it with ATP. Okay? And then here I drew a hydrogen, a negatively charged hydrogen, which is called what? That's a hydride, and that's being donated by the NADPH. That hydride is going to attack this number 1 carbon. The phosphate leaves, and now you've created glyceraldehyde 3-phosphate, which can be used for glucose synthesis. Okay? Very simple. Okay, here's where it gets a little complicated. Okay, so here's where it starts to get a little complicated. I'm going to try to make this as simple as possible. So this is a little more detail about the Calvin cycle. Okay, so here it actually requires quite a bit of ATP because you have to make a lot of these molecules of this product from Rubisco. Okay, I'm sorry, this is the product from Rubisco. These 3 phosphoglycerates. You have to actually make 12 of these. I don't think you need to memorize this, but all I would like you to know here is that some of these glyceraldehyde 3-phosphates, some of this stuff, actually has to be recycled to make the acceptor molecule, this RU5P molecule. And some of it actually goes on to make glucose 1-phosphate, and the glucose 1-phosphate eventually gets incorporated into amylose. Okay? So altogether, it requires 19, I wrote down here, 19 ATP molecules, 6 plus 12 plus 1, in order to incorporate 1 glucose into amylose. Okay? This is a polysaccharide. This. By the way, this 3-FBP, that's fructose 1,6 bisphosphate. Just to let you guys remember, that's also part of the gluconeogenesis pathway. Okay, we're now ready to talk about a summary of the light reactions and the dark reactions. Okay, so this is on page 699 of your book. The dark reactions, we're going to have carbon dioxide being incorporated into glucose. That requires 12 NADPHs, 12 protons, and we're going to incorporate those carbons into glucose finally. And then this requires 18 ATPs plus water to be hydrolyzed to ADP and inorganic phosphate. So those are the dark reactions. And this is a summary of the dark reactions right here. And then we have the light reactions. Okay? And then the light reactions, remember, we're trying to make NADPH and we're trying to make ATP using sunlight, right? And so we've talked about how water gets converted into oxygen. We talked about that in the Z scheme, and we've talked about how the NADP plus gets reduced to NADH. That's also part of the Z scheme. And we talked about how ADP can be converted into ATP through the proton motive force, which is created across the membrane of the thylakoid. So I'm trying to put together all the different pieces that we've talked about over the last few days. Okay? At the end of the day, if you summarize the dark reactions, if you sum up the dark reactions and the light reactions, okay, you're going to get this summary here, where you get 6 water molecules plus 6 CO2 molecules, 48 photons, and you're going to create 1 mole of glucose and 6 moles of O2. So I know you guys have seen this probably in basic biology, but now you can appreciate the details into how this all comes about. It's not just a simple, oh, you just zap this thing and you get these O2 molecules out. It's a fairly lengthy and complicated process. Okay, let's just get started on one of those problems that I was going to go over with you. And so this is problem 1, where it says, according to figure 612b, upon excitation of the P700 reaction center, upon excitation, the P700 reaction center is raised in potential from plus 0.4 volts to minus 0.13 volts. To what value of standard free energy change does this correspond? And how does it compare with the energy of an Einstein, that's a unit of measurement, Einstein, of 700 nanometer photons? So I think I did this problem. Yeah, I did this problem. And I'll set it up how I think about it. So problem 16.1. Let's get down the equations, right, that we're gonna need for this. So for sure, the first part, let me just go ahead and so the first part is when I see that delta G and I see volts, I need to come up with an equation that converts the volt difference to a delta G value. So that's going to be delta G under standard conditions is going to be equal to minus NF delta E. Oh, sorry, that got so big. There we go. Okay. And I know that delta E under standard conditions is going to be equal to something minus something. Does anybody remember what should be the subscript here and the subscript here? acceptor minus donor, right? Okay. And the two volts that we were looking at was 0.4 volts and the other one was minus 1.3 volts. Okay. So in this case, the acceptor is going to be the 0.4 volts, and the donor will be the minus 1.3 volts. So let me go ahead and draw this nicer for you. Delta E under standard conditions is going to be equal to 0.4 minus a minus 1.3. How do I know that? I know that because anything more positive is going to be an acceptor, okay? And anything more negative is going to be a donor. In order for this to be an exergonic reaction, for this to be an exergonic reaction, I know that the acceptor has to be more positive than the donor if this is going to be an exergonic reaction under standard conditions. Okay, so that's going to be simple then. Now I have all the information that I need in order to figure this out. Okay, and it turns out that yeah, it's going to be just one electron that's going to move. It's going to be moving through this 96.5 times 0.4 plus 1.3. And so I'm going to get from this 164 kilojoules per mole. Okay. Next, we have to figure out the second part of the equation. We have to figure out how much energy we get from an Einstein of 700 nanometer photons. Okay, so I'm going to write that down, 700 nanometers, so, and one Einstein. One Einstein of 700 nanometer photons. Physics, energy is going to be equal to Planck's constant times, I think that's the frequency, which is going to be equal to Planck's constant multiplied by the speed of light divided by lambda. Okay. And here I have all the information I need. I know that that's a constant. That's going to be 3.0 times 10 to the 8th meters per second. Planck's constant is 6.626 times 10 to the minus 34 joules multiplied by seconds. And this, lambda, is given as 700 nanometers. So we just have to make sure that our units are going to cross out correctly, right? So that energy is going to be equal to 6.6 times 10 to the minus 34 joules times seconds times 3 times 10 to the 8th meters per second times 6.02 times 10 to the 23rd photons per mole. Okay. Multiplied by 10 to the 9th nanometers per meter, multiplied by 1 kilojoule per 1,000 joules, divided by 700 nanometers. Okay. Let's cross these things out carefully. Meters, seconds, joules. Oh, nanometer photon. Okay. And so now we get, if you do this calculation, you get 170 kilojoules per mole, which matches up pretty well with the 164 kilojoules per mole that you got from the from the delta E. So that's the answer to the first problem. We'll do the next two next time, and then we're going to get into chapter 17 and talk about fat metabolism, which is very exciting, actually, very important medically. Okay. Hello. I also want to say that I forgot a minus sign right here, so I'm going to put a minus sign. I put a minus sign right here. All we're trying to calculate, though, is if the energy in an Einstein would be sufficient to supply the energy for this reaction. So that's all I'm trying to do. Okay, I'll sign off here. Bye-bye.